So, you know, it's a pleasure for me to be here again. And the again is like, makes me know how old I am, because I was talking to Rich Schukel, we think it was like 1987 that I was here. And it's sort of like when I was getting my career started. And indeed, work on the Hessian fly is what got me rolling on a lot of this modeling and things that I've been doing for most of my career. And what led me into what I'll be talking to you about today. So I've had a great visit here so far, and look forward to the rest. And so, pretty big topic here. Genetic engineering, you know, is something in agriculture as well that is something people are talking about a lot. And I think probably quite a few of you know about genetically engineered crops. And this is transgenic host plant resistance. And probably many of you have seen these kinds of slides. This is conventional corn in North Carolina. And this is transgenic corn that has a gene from Bacillus thuringiensis expressed. And this is damage from the corn earworm. And you can see that you don't see the damage on the other side. And this has been out in the field since 1996, but it isn't as if everyone thinks it's a great approach. And I think that you know that there have been a lot of people who have protested having this, that in Europe it's not something that's really well accepted. And there's even right now some pushback on this in terms of having all produce products that have genetically engineered crops as part of their components being labeled so people can choose whether or not they want to buy these kinds of products. But this is genetically engineered host plant resistance. What I want to talk to you about today is not this, but more genetically engineering the pest itself as a management tool. So what we'll be discussing are insects that have been genetically engineered. And many of you, are in entomology, so you've probably seen things like this. So this is Aedes aegypti under a fluorescent microscope, and you always have a little bit of autofluorescence in organisms. But this one was genetically engineered by Alexander Franz, and what you're seeing here is a marker gene in the eye, and this is green fluorescent protein being expressed. But what's more important, more interesting, in Aedes aegypti, this is the insect that transmits dengue virus, is that in the midgut you're seeing this GFP, green fluorescent protein, being expressed. And this is a gene that's turned on for production of an RNAi towards one of the genes in the dengue virus. What's really interesting in the way that they engineered this is they borrowed a promoter that only turns on a gene in a female after she's taken a blood meal. So this is a picture of a female that just took a blood meal, and now that gene has been turned on. And what it does is it silences the virus gene so it can't replicate in the insect and can't be transmitted. All right? So this is what I mean by use of genetically engineered pests. All right? So you can imagine that with something like that, where there's that much controversy and that much biology involved, you can't just do the molecular genetics. You need to be dealing with the molecular biology, the population genetics of how are you going to release insects like that and have them actually take over a population. You need to understand a lot about the ecology. But because this is something that society is very interested in about, and because you're developing a product, you have to bring in other areas as well. So this immediately becomes interdisciplinary kind of research to develop these kinds of things. And I'll be getting into talking about that as we go on. But what I want to say is this is not going to be a talk about just my research. It's just to introduce you in a way to this area of research. I find typically when I come and talk, even at entomology departments, a lot of people haven't really read very much about these approaches. So I think it's useful to introduce this. What I'll be doing is first explaining the concepts behind transgenic pests and invasive organisms for their own management. and I'll talk about potential impacts of advances in molecular biology, and then I want to describe a little bit this new program that we have at NC State that Steve was talking about. So to go back to the general introduction here, there are two general approaches to using genetically modified insects for their own control. One is to use that for suppression or local eradication of an insect population, and the other is to replace a pestiferous or virulent strain of the insect with a more benign form or more benign genotypes of that pest. For example, that mosquito that can no longer transmit dengue. So suppression or local eradication is also called autocidal control. And one of my graduate students was on the web and found a slide to sort of explain this to people in just a much more convincing manner. And this is exactly where we're going. Right? There's a Chinese advertisement. But I mean, if we, some molecular geneticists think they're really very good, but we don't think we could get there. But anyway, to go back, this is where you might want to go. Genetic engineering is one of these things, but this is not something brand new. This is something actually an old concept, an old approach. And this is a picture of Edward Nippling many years ago in the 50s when he really sort of brought this to the fore, the use of radiation to sterilize insects and use a sterile insect technique. And this was sort of the forerunner to what we're dealing with right now. And for this work, he won the 1992 World Food Prize for his contributions. And I'm going to go through some of the history to bring us to the point of where we are today. So basically, the idea behind the sterile insect release technique was whenever you see green here, that's going to indicate just the normal insect genotype, and red is an insect that has been modified. In this case, with radiation, they've been modified by the male being exposed to a dose of radiation that causes chromosomal breakage. Chromosomal breakage doesn't mean that their sperm won't be viable. What happens is that the male can fertilize a normal female, but during embryogenesis, there's malfunction, and you either get dead or sterile offspring. Okay? And I'm assuming that all of you have heard something about sterile insect technique. So the idea is if you can produce enough of these and flood the population, the normal population, with lots of males that are sterile, so they outnumber the normal males, then if this is a 9-to-1 ratio and they're mating with these females, you'll wind up with 90% dead or sterile offspring. And if you keep doing that, so let's say you had a local male population of 10,000 males, and this is the population density on a log scale, so that's 10,000, and every generation you're releasing 90,000 of these males in that originally 9-to-1 ratio, the population goes down steadily and faster as it goes down, because what's happening is that 90,000 in the beginning is a 9-to-1 ratio, but as the population is going down, that 9-to-1 becomes 90-to-1, becomes 900-to-1, so you're having even more and more effect. So it's actually called inverse density dependent effects here. So this was very successful with the screwworm fly, and because this is an entomology one, I decided to show you some slides of this actual process beyond all of the conceptual stuff. You want to see how this actually works. So what was amazing is that what they were able to do was release enough of these flies, starting in everything in green was the geographic distribution in the summer of Hessian flies in the 1950s. And this is just a summer range where they can blow up very far up into the Midwest, including your area, I guess. So they started in Florida on Sanibel Island, releasing these irradiated males. And all the green now has been areas where that Hessian fly has been eradicated. So it started off eradicating it all the way down to the border, but this border actually became indefensible because flies were crossing the border and reinfesting. And that led the USDA to come up with arrangements with the Mexican government to keep that program going. and push them down through Mexico, and more recently arrangements in Costa Rica and in Panama to move them all the way down. All right, and right now they're maintaining a boundary in Panama. This shows a picture of the original factory for production of the irradiated Hessian flies. And you can see this, there's no Priuses out there. That was a long time ago. These are 1950s sedans and such. And this is where things started. This shows a picture inside that factory where they're raising the screwworm flies. This is on artificial media that they're raising, and these are the larvae, and when they finish feeding, they work their way out and into these troughs, and then the pupae are irradiated, and then the adults are dropped out of airplanes. So this was the old factory production, and I recently got the opportunity to go down to Panama to the new factory. This is the new USDA Panama screwworm facility near Panama City. And it's a beautiful new facility with administrative buildings. And then going in here, everybody has to wash down, put on suits. It's very official and very impressive. They're producing between 60 and 120 million Hessian flies per week. I didn't figure out what that is a year, but it's a lot of Hessian flies. And just to give you a sense of what it's like inside here, these are the adult cages where they lay eggs. And this is a close-up on those screens. You can see the flies themselves. And interestingly, the substrate, right, these lay their eggs on cattle. That's, you know, what they're pests of. But they have them laying eggs on these boards that are just heated and rough. They collect the eggs. These are the egg batches. And they put those egg batches on media, on food. Oh, this is their little blender for blending that food. This is a huge enterprise, and this is the batch of eggs on the media that they use to raise it. I don't know all the ingredients in it, but once those eggs hatch and the larvae come out, it looks a little bit messy. It smells a little bit messy, but that's the way the production goes. But as the larvae get old enough to where they're finished feeding, they work their way out of these trays and start coming out. And this is all set up that way in those factories. This just shows you the stacks and stacks of these trays. And what's interesting is when they fall out of that, there are these little troughs on the edges of the concrete. And when the larvae fall into that, they can't get out. And so this is a picture of those larvae. And they just come in and scoop them out and put them in with sawdust, and they pupate. And it's these pupae that are then brought into the radiation room, radiated, and then they put into airplanes. So anyway, I thought for an entomology audience to show you what goes on there. This is a huge infrastructure, right? And the USDA likes the idea, but they're actually torn about the investment that goes into this, even though it saves something like $1.2 billion a year from Latin America and in the US to the farmers. All right, so basically this worked fine for the Hessian fly, but it has not worked in a lot of other pests. And I just want to go in just a little bit of the detail. It works fine if you have this inverse density dependence, but you don't have anything else pushing up. And any of you who've taken insect ecology know that as you decrease the numbers of pests, you also reduce the percent parasitism typically, because parasitism typically is density dependent. And a lot of other mortality and a lot of competition is density dependent. So if you have the right ecology, this approach can work fine, but if not, you might wind up where you're pushing them down and other actions are pushing things up against you. So it worked with the Hessian fly, but it might not be the best approach always for everything else. One other thing to mention is, of course, when they were doing this is in the 50s, they didn't even have an EPA or anything like that. People didn't have to worry about off-target effects, and we'll get into some of those details as we move on. But the issue is this was a huge amount of investment, even for the Hessian fly, which is actually a very rare insect. Actually, there's not very high density like you would have with aphids or some other European corn borer or something like that with hundreds of thousands per acre. So the question is, can you improve the efficiency and guard against environmental impacts compared to this approach? And so you start with this idea that you're having to release 9 genetically modified insects per normal insect. And a question would be, could you turn that around where you were releasing 1 genetically modified insect per 9 of the normal insect? So basically, could you improve this 100-fold and thereby make it more accessible for other insect species and more affordable in general? So this is a picture of those, a relative of the screwworm Lucilla that was made in one of the labs at NC State recently. I don't know if you know, NC State colors are red and white, but this wasn't done necessarily for that reason. This is work done by Max Scott, and the red-looking insects are the females, and the white ones are the males. This is done with sex-specific splicing and just expression only in the females, and those insects will then die before they pupate, and you'll only be producing males. I said that you're supposed to be just releasing males according to nippling and all of those approaches, but with the screwworm fly, they've never been able to separate the sexes, and they actually release males and females. It does work, but it's a lot less efficient. So this kind of a technique would enable you to just be releasing the males. And this is just a preliminary thing about what can be done later with the transgenics in terms of improving this efficiency. All right, so I said there were 2 kinds of general approaches, and we talked about the suppression, local eradication, and the other one is this idea of strain replacement. So basically, in this case, you're trying to take this green insect that's causing problems and turn it into a more benign insect through genetic modifications. So the idea here is that you would be able to release these insects into the wild. But if you think, wait a minute, you build this transgenic or insect and it can't transmit disease, and well, let's say you have release 1,000 mosquitoes that can transmit disease, but wait, there are a lot of them that still can, right? So that wouldn't really do too much in reducing dengue. So what you really need to do is be able to put that gene out and have something about it that will cause it to replace all of these other insects, so that in the end you wind up with all of your insects not transmitting the disease. All right? So we'll get into talking about that approach. So this is, you know, sort of one of those things that I just showed you about the insect itself. And I'm not going to actually go into the construct and everything that has the double-stranded RNA and everything, but that makes the insect not be able to transmit. But it doesn't, and it could have a fitness cost, right? Those insects might actually be less fit than the others. How are you going to ever get that into the entire population? So what I want to do is talk about gene drive and how to do that. But I also want to emphasize that the concepts for this are, again, nothing new. It's not something that people talk about typically in entomology departments or entomology classes, but back in the 1940s, a fellow named Vanderplank was working with tsetse flies, and he came up with ways of hybridizing those tsetse flies so that he could get one strain to replace another, and I'll talk about that a little bit more. But while this Brit was working, there was also Serebrovsky working in the Soviet Union on approaches of using translocations to do that same kind of replacement, or that kind of gene drive, that would drive genes into a population, though they were also considering use of these things for lowering population size. All right, and it's very interesting in these whole areas. Vanderplank never published some of his results. They were only published in 2005, after his death. Apparently he would have lost his pension if he had published that for some reason. At least that's what his son told everybody. And Serebrovsky was working in the Soviet Union during the Lysenko era, when Mendelian genetics were outlawed for about 40 years. And he was just lucky that he stayed alive, actually. I mean, some of his compatriots were jailed. So this stuff was going on. Serebrovsky's work was never disseminated widely, and he did this in the 1940s. His work was redone by Chris Curtis in the UK in the 1960s, and once Chris Curtis published his work, the Soviet Union released the work that was done by Serebrovsky. There's lots of interesting history here, but I just want to give you this idea that the concepts behind this are not new. But I do want to tell you what did all these three people were working on in terms of these systems. And what they were working on was a phenomenon called underdominance. Many of you know what the genetics is of dominance, right, where the offspring resemble one parent. Some of you also know about overdominance, where the heterozygote has higher fitness than either of the parents. Underdominance is when you would be mating two individuals. This is fitness on this scale, and you have big AA and little AA. You mate them, and the offspring are less fit than either of the parent. And you notice in this slide, this green one is more fit than the red one, okay? And if what you want to do is replace the green one with the red one, because the red one is not as virulent, how could you do that? But there are interesting population dynamics here that actually whenever you have a system like this, there's only two stable equilibria, and that's fixation of the green or fixation of the red. And the dynamics look like this. If you look at, and this is for a translocation dynamics, where there's just this kind of underdominance. And if both of these were equal, there's an unstable equilibrium point where if both of them are exactly 50% genes for the green, 50% for the red, they could stay at that 50% level. But if they move away from that unstable equilibrium, either up or down, they go all the way to fixation of one of the two types. Either the translocation gets fixed or it's lost, okay? And so it's an interesting dynamic that if you could introduce this red insect, you know, the one that can't transmit, above that 50% level, it would go to fixation. It would push itself into the population. All right? So, why? All right, so it's an interesting, so I have this, I hope this helps. So if you think about this population, right, where you had this green population that's transmitting disease, and you release this whole mess of red ones for a short period of time, and you only do it once, unlike the sterile insect technique where you have to release every generation for a long time to get rid of them. You only release once. What's going to happen here, if the two of them are compatible and they don't discriminate between each other in terms of mating? And that was what Vanderplank found with the tsetse flies, that they would mate randomly with each other. So if we look at this a little bit closer and we focus just on one of these red insects, and we say it's going to mate with the nearest neighbor, you know, just to give you the simplest kind of approach to it, they would have a chance of 2, 3, 4, 5 out of 6 matings, right, randomly. So 5/6 of their matings would be with a like type. And what I just told you is if they mate with a like type, then their offspring would have this fitness. But if they mate with the green type, their offspring would have lower fitness. So in 5 out of 6 cases, they'd have that fitness, and in 1 out of 6 cases, their offspring would have that lower fitness. All right, so having the other type around is problematic, but only 1 out of 6. So now if we look at the next slide and we focus on one of these green individuals, right, it's mostly surrounded by the red individuals. So instead of making 1 out of 6 bad choices of mate, they're going to, on randomly or probability-wise, make 5 out of 6 bad choices. And even though when they mate with themselves, they even have higher fitness, right, than these mating with themselves, 5 out of the 6 matings put them down here. So the average is lower fitness than these. And so that's what drives this system, is that as long as you're in the majority, you're making fewer mating mistakes, right? Your offspring are having higher fitness, and as you become more frequent, you make even fewer mating mistakes, and the other type is making more mating mistakes, so it goes up to fixation. So that was the background behind this underdominance. And this was the first approach, basically, that could lead to this kind of gene drive where you put some genotype out there, and not only does it get out there, but it actually leads to it being driven to fixation, so you wind up with a whole population that's not transmitting the disease. and might be less fit. And there's a lot of work on this back in the 60s and 70s, and a big project led by Chris Curtis and others in India. And this just shows you this great picture of this bicycle rickshaw where they're releasing mosquitoes out of the back. And this was going on for about 12 years before there was an article in Indian media. It was a newspaper at that time saying that this was actually the West conducting biowarfare on the Indian people, because some of these were done with chemosterilants. And that actually, they were working with WHO, which refused at that time. Their policy was never to respond to these kind of crazy media things. And the bottom line was that the media just shut them down completely, and the whole program was stopped, which was really unfortunate. It was great work being done. But 10 years later, after more work at this time, Chris Curtis, who you saw his picture, was doing a lot of this, wrote an article calling the whole thing a very interesting lead balloon. And you want a balloon to rise, not a lead balloon. There, out of all the stuff I'm telling you about, there were a lot of efforts put in there and a lot of really smart people working on that. But the techniques that they had at their disposal seemed to hold things down. And that's what he was talking about with the lead balloon, was we had a lot of great ideas, but when they were trying to make these translocations and all that, the fitness effects of some of these things were just too strong. And I do want to emphasize that the people who are working in this, if you read their papers, we have a class in genetic pest management, we have the students read these things. They are thick, they are dense. The approaches that they use, the manipulations they did with the pests, not only the conceptual but the empirical work, really was painstaking to use classical genetic techniques to get these things to happen. So the rest of this thing is going to be a little bit about, is molecular biology to the rescue. I don't know if those are exclamation points or question marks, so we'll talk about that. So, just when all of this is happening, the first transformation of Drosophila starts going on, just about the year after Curtis published that paper. So now we have more genes, more precise genomic manipulation, and more powerful gene drive mechanisms. But you have the same ecology, right? A pest that has very strong density-dependent population regulation isn't going to change just because you say you're a molecular biologist, all right? And also, we have more social concerns than when they were doing the other work. So, to just go back to the picture I gave you, when we were talking about the classical approaches, you basically had this sterile insect approach, and you had translocations and these interspecific underdominance approaches, both underdominance approaches that could work. Today we have something, this is just a short list of different approaches that are accessible in these two kind of realms, and some of them go across this. So I'm not going to talk to you about all of these, but I will just touch on a couple of them just to give you a sense of what's being done. So first, to talk about homing endonucleases. Homing endonucleases can actually be used in either of these kinds of approaches. So what is a homing endonuclease gene? It is a gene that's found in bacteria, in yeast, and some other organisms, but had never been found in diploid higher organisms like insects. But Austin Burt felt he had a great idea and that this could serve as a gene drive mechanism, and he had some ideas about it. And he got the Gates Foundation to give him like $9 million to get a start on this. And he got this great team together and they started working on it. But let me tell you what the concept is behind this. So this is showing a nucleus and a nuclear envelope, and assume just an insect that is diploid and only has one chromosome. And on that chromosome, you have a homing endonuclease gene inserted into one of the homologs, but not in the other. So this is like a heterozygote, right? Homing endonuclease means that it's a nuclease that breaks, that cuts DNA, and it homes in on a certain spot in the DNA, all right? And this one is right here in this spot, and this is the homolog, and this is the sequence that is the recognition site for the homing. So what you see here is, in this case, where it inserts, it breaks up that site, right? So this is the recognition site. Once the homing endonuclease gene is inside there, it's broken that site up. So what's happening in a heterozygote is that the homing endonuclease gene codes for mRNA, goes back inside and makes homing endonuclease protein, which then homes in on that recognition site that's in the one that doesn't have that. It can't affect this because that site has been changed, right? It homes in on that, and as a nuclease, it cuts that DNA. And all organisms have DNA repair mechanisms. And so what the DNA does in most cases is to use the template here to repair that DNA. And all of a sudden you've gone from being a heterozygote to a homozygote. So if you think about what happens here, if you think about typical Mendelian inheritance, and you all know this from high school, right? If you had a heterozygote for the homing endonuclease gene mated to a normal individual, right, half the offspring would get the homing endonuclease gene. But if this thing duplicates and make this thing during meiosis into homozygote, both of the F1s get it. And I like to call this super Mendelian inheritance, all right? Because you're inheriting more than that 50%. So what's the dynamics of this? The heterozygote, if you had this placed in a gene, you were breaking a gene in half that was important to function of an organism, right? Where that insertion was, right? So this is a functioning gene, and it has that recognition site. When the homing endonuclease gene inserts, not only is the recognition site broken up, but that gene has this big extra sequence in it, and it can't function. Now, so there are a lot of recessive lethals. I wrote up cystic fibrosis is one of those things, right? If you're a heterozygote, you can do fine, but if you're homozygote, you can't. So the heterozygote does fine, the homozygote will die, all right? So if the dynamics of this is if this is homing endonuclease frequency, if you just release a very small fraction of individuals that have the homing endonuclease gene over time, and I should say time over here, it will increase to a point where you could be up to 90%, not at 100%, 90% that will have the homing endonuclease gene. And let me talk to you about the dynamics here. And I'm not going to give you the actual equations that are used for this, but just to bring it back to just Hardy-Weinberg equations, right? I think most of you know p squared plus 2pq plus q squared is equal to 1, right? That's all the three different genotypes. But when you have a p of 0.9, the normal individual's frequency of 0.9, and the homing endonuclease is just 10%, right? And that's where you're starting out. If you try to figure out how many homozygotes you have and how many heterozygotes you have, basically you're going to have mostly homozygotes for not having the homing endonuclease. It's rare. You're going to have 18% 2pq heterozygotes, and they're going to do fine. And then you're going to have 1% homozygotes, and those homozygotes are going to die, right? So what's happening here is you keep increasing. As long as you have a heterozygote, those are going to turn into meiotic events that are going to be like homozygotes, going to lead to the next generation having more homozygotes, and your frequency is going to start going up. And that's what happens here. And there's a balance between how often you have this homing and how often you have, what percent you have mortality. So once it gets up to that higher frequency, and I'm assuming this is 0.9, you're winding up with 81%, right, 0.9 squared, are going to have the homing endonuclease in homozygote form, and 18% are going to be the heterozygotes. And what's happening is those are all dying. And that becomes actually the equilibrium point. And that means that when an individual has offspring, 8 out of 10 offspring will die. So basically you have a population with 80% drag on the population dynamics. And what's basically happening is, so you have pressure due to that super Mendelian inheritance to push the gene frequency up, pressure by mortality of the homozygotes, and that gets stronger actually in mid-frequency, but once you get up to high frequency, those two balance out. But in the meantime, you're having a lot of mortality. So, $9 million later, the team working on this, including Austin Burt, actually engineered this into Anopheles and showed that the frequency, the fraction, would go up as predicted. This is still very early days, but it's just sort of proof of principle that this kind of thing could happen. All right. So I just want to give you a little bit of a smorgasbord of, the stuff that people are playing with. Another one that is being used more for gene drive for that strain replacement is what's called a Medea element. And Medea elements were first discovered in tribolium beetles as a natural kind of occurrence. Okay, the name Medea basically comes from the Greek or Roman goddess Medea, who is a god. I'm not sure if she was a goddess, but she was a figure in Greek mythology who killed her children to spite her husband. Okay, remember that. Okay, so this is a female tribolium beetle with Medea element on one of the two homologs. All right, when she undergoes meiosis, you get one in each time. If she mates with a male that doesn't have it, half of her offspring get the Medea and half don't. It's not occurring like that super Mendelian inheritance with the homing endonuclease. The embryos actually are in a 50-50 ratio. But the interesting thing is any individuals that don't inherit her Medea gene die. So if they look too much like the husband, out. So I think that's where Dick Beeman came up with this name, not my idea. All right, so you have that, a little more detail on it. What the idea behind it is that the heterozygous, during gametogenesis, you have a substance laid down by this gene, the Medea element, and that substance is not toxic to the gametes, but it's toxic to embryos. Embryogenesis. So when the gametes are formed, they're fine. Mates with a father, a homozygote that doesn't have it, form embryos that both have this substance that kills embryos. But when you receive that Medea gene, it has a second function, and that is to detoxify the toxin. So this one gets rid of the toxin. It has an antidote. And this one does not, and it dies. There's a lot, Marcel Lorenzen at NC State has actually isolated this element and cloned it and is trying to figure out how the hell it works. But in the meantime, Bruce Hay and his group at Caltech, he was working on apoptosis. He said, gee, you know what? I can build this. And so he did. And he built a synthetic form of Medea, and was very interested and did this in Drosophila. Basically what he did, he built a construct that had this maternal toxin, right? He had basically the maternal toxin, a promoter that was only turned on during gametogenesis, right? That produced that and laid down that toxin, and then the other component working in the opposite direction was a zygotic antidote, and this was actually microRNAs that were functioning here, and here a separate form of the gene that was going to be silenced, but with different DNA coding so that it wouldn't be silenced by the microRNAs. So we had both of these components so that if you produced that maternal toxin but you inherited this part, you would have an antidote for that toxin. And he did experiments where if he put the Medea in a male, mated it with a normal female, you got 50% each time and you got very high survival. If you did it the other way around, where the mother was the heterozygote, the father didn't have it, 48% survival, almost perfect. And he took this, and this is why it was published in Science, of course, he took this and he actually put it into a population of Drosophila at a frequency of about 0.5. And this is looking at the wild type individuals, and you can see here, this red line was the model, the population genetic model of what should happen, and these are the replicates. So pretty nice in Drosophila. He's been trying to get this into mosquitoes. It's not so easy, but that's where things are moving towards. So one of the things that you can recognize here is there are issues of regulation and such. And if you think about this kind of a pattern, you see that you go from population with a low frequency and that the Medea element takes over. But there's actually nothing to stop the Medea element. So it's something like, if anybody ever read Kurt Vonnegut's book, it's like Ice 9, you know, where just a little crystal of it and it just keeps going. So where it doesn't recognize borders. So if Kenya decided it wanted to put this into Anopheles and Tanzania did not, it's not going to recognize borders. It also might not work. Let's say you got that RNAi into the population and then you find out, oops, it only works for certain genotypes and it's not really live that long. You can't, it's hard to retract what you've done. And you can imagine regulatory agents don't like that. So we came up with a different approach. Sort of, those little Post-it notes that you use. Somebody came up with that. Basically, there was a glue that they had at DuPont or something and didn't work very well, and they were going to put it on the shelf. And somebody said, wait, I like this glue. It's not very good. It's important, and that's why you have Post-it notes, right? So we thought, wait, we need a gene drive system that's not very good. So this is what we developed, is what's called a killer rescue system, which is much simpler than these others. But the idea is that it would be temporally and spatially limited. So you could put it out there and it would increase in the population and then it would decline. So you could test something out and then try it again if it wasn't working very well. So it has a very simple approach. Here I'll show this in a diagram. I'm going to deal with Lepidoptera here with 4 wings. All right, so here the red means that it has a gene for that change. Right, so this is a killer gene. Right, so it's homozygous for that killer gene. This sort of representation. But it also has a rescue gene, sort of like with that Medea element, but it could be on a different chromosome and it can act any time during development as long as the 2 canceled each other out. And you put an anti-pathogen gene in tandem with that rescue gene. So you wind up with an individual that's homozygous for the killer, homozygous for the rescue, and that individual will be happy, right? Because it has both. So if you have an individual that has both, mates with a female that doesn't have either one, you get one that's heterozygous for both, and that's happy, because these are dominant acting genes. But when that heterozygote then mates with the female, or it could be going both ways at this point, you wind up with a different situation. These are the F2 offspring, where they're all segregating independently because they're on different chromosomes. And what you wind up with is some have the killer and the rescue gene, some just have the rescue gene, some just have the killer gene. And if you arrange that, so actually the killer gene is only killing individuals here that only have that killer and not the rescue. So the ones with the rescue or the normal ones are not dying. So if you look at that in terms of the population genetics, you're knocking out these individuals that have the killer, but the rescue gene, any ones that have the rescue and the killer, are maintaining themselves in the population. So if you count all of this up, what you see is that after the killing, instead of having 50%, right, let me go back for a second. All right. If you count these up, each of these alleles for the killer and the rescue are at 0.5, because that's what you released. But after the killing, you're going to wind up with 8 out of the 12 survivors having the rescue gene. So it went from 0.5 to 0.66, right, because you've knocked out all these individuals that don't have the rescue gene. But after the killing, only 4 out of the 12 offspring are going to have the killer gene, because whenever the killer gene was alone, it got knocked out of the population. So basically you're having this push-pull kind of thing. And if you graph this over time, what you see happening in terms of frequency, if you release these at about 0.33 is the initial gene frequency, the rescue gene over time, this is 120 generations, rescue gene is pushed up close to fixation. The killer gene is slowly lost from the population because each generation is being pushed out. The wild type is pushed down to almost 0, because this is the gene frequency of the rescue, and if you think about that in terms of Q square, you have very few wild-type individuals. But over time, it's coming back up into the population, because once the killer gene is lost from the population, there's nothing maintaining the rescue gene, and if there's any kind of a fitness cost to it, it declines in the population. And the whole idea behind this is you could get about 40 generations in which you could test your anti-pathogen gene to see if the thing is working and work that in a local situation. So, I'm not sure how I'm doing on time here. When's the seminar end?
You've got about another 10 minutes?
Another 10 minutes, okay, very good. So this thing, you can see all of these ideas and stuff like that are generated, but you know, is this going to be something really worthwhile? So what you're seeing so far is this molecular biology and populations going together. But when somebody just tried to release, a small company, these genetically modified insects just for having basically SIT, there's a really big response in the community. And I'm not going to go into all of the detail, but just to say people are very concerned about you releasing genetically engineered mosquitoes. One thing that we've been doing, and I'll go through this kind of quickly, is that that you really need to look at the ecological part of this. And we've been working in the city of Iquitos, Peru, where they have dengue and have these mosquitoes, to sort of model all of this and see if it could really work. And we've been working with a team that's been collecting ecological data for many years. And these are two of the people who have been building these detailed models of Aedes aegypti and the genetics of this, so that you're not just working with these simple models that I just showed you, but actually looking at models that include individual houses, include the movement of mosquitoes between houses, all the structuring, and then looking at a lot of the ecology of the mosquitoes. These are mosquitoes that breed in containers and tires and such, doing detailed analysis of the density dependence in the system to figure out which of these methods will work. And we found some very interesting counterintuitive results in some cases where things would work or not, and also approaches for how you would actually do the releases. You know, if this is just a grid of the city, do you release a high percentage of them in the center of the city and let them move out, or do you release them more broadly and see what happens? Sometimes this will actually implode on itself. So it's a utility of doing more detailed analysis with the ecology. But I don't want to go into too much, but to say that you're going to have to do these ecological studies if you can get through regulatory. But the other thing was, when talking to you more about these engineered things for health reasons, but this question about sustaining biodiversity that was in the topic. So the idea is that you don't have to have a target system that's just mosquitoes. Targets could be, for humans, there's also bird malaria, agricultural pests, and other human problems. I just want to focus here quickly on the Hawaiian honey creepers. Right? You're out on an island. You've been there for millions of years, that's where you've evolved. There's no malaria out there, bird malaria, because there's no insects to transmit it. About 100 years ago, Culex quinquaciatus arrived in Hawaii and has been transmitting bird malaria. The non-native species of birds that have come to Hawaii are resistant to the malaria, but the indigenous birds are not. And a lot of these are on the verge of extinction, and with global climate change, they're right now up on tops of mountains and stuff. There's concern, right? So could you actually do engineering to make this mosquito not be able to transmit bird malaria and help save the Hawaiian honeycreeper biodiversity? A lot of other places where that could come in. We're working with island conservation. One of the things on islands that's a real problem are mice and rats that actually decrease bird biodiversity on islands. And they get rid of these mice, cause extinctions, by flying helicopters over the islands and dropping rat poison. It's successful, but it's very controversial. Tsetse fly is my counterexample in a way. So tsetse fly is important because of trypanosomiasis, which is a problem for cattle and also a problem for humans. And this is the zone where the tsetse fly exists, and a lot of people talk about, wouldn't it be great to get rid of tsetse fly? This is also one of the zones of highest biodiversity in the world, right? Because of the tsetse fly, this is underdeveloped area that has very high biodiversity. What happens if you open this all up to agricultural practices? And people have done analyses of what would happen if small holders were in the fields versus large landholders coming in, and what would happen to biodiversity. Definitely go down. So you have this kind of a thing about helping human populations, helping biodiversity. Would the development in these countries be something that would actually help bring people out of poverty or make it worse for them? You've seen what's happened when oil is found in specific countries. So the issue is, of course, that we need to deal with these ecological issues and the molecular issues. But at NC State, what we decided was we needed to broaden ourselves. We had this program going, but we brought in people from other areas, other colleges, university, and tried to pull them together. And we wrote a grant to the National Science Foundation called Genetic Engineering in Society, the Case of Transgenic Pests. And the idea is to take all these different foci and look at this problem from different perspectives and see how to be interacting. And it's a graduate program. We brought in our first 6 students to work on Aedes mosquitoes. Next year we're bringing in a cohort to work on mice with that problem on the islands. And in the third year we're going to be bringing in people to work on agricultural pests. So if there are any students, I said this before, there's a little advertisement, who are looking for PhD programs, we're going to be recruiting people for the year 2014 to be working on agricultural pests. And I'll just end with the fact that, the idea is, if you look at most graduate programs, it's learning more and more about less and less until you know everything about nothing. I think some of you have heard that, right? So the idea of this program is that we wouldn't be doing that. We'd be sort of giving people backgrounds in a variety of things and educating them in some sort of central area of this circle. Not too much in the middle where you're too broad and you can't get depth, but we're trying to find a balance. And that program is going on with the idea that we focus on one system and then we look at it from these different perspectives with graduate students who are in different departments. So that is a program that we're starting and trying to address these things, and I will leave it at that. Thank you very much.